So thank you very much for the introduction, and thank you also for the invitation to come to this meeting. This is my first time at a JGI meeting. It's been really enjoyable, really interesting for me. So I'm going to talk today, I'm going to kind of switch gears a little bit from a lot of the talks that we've heard so far and talk about automation, but talking about it from a sociological perspective. So a little different from most of the talks that we've heard, but I'm hoping there will be some resonances, perhaps resonances for those of you who work in facilities that make use of a lot of automation, or those of you whose projects depend on the increasing use of automated tools. So the work that I'm going to talk about today comes from a project funded by the European Research Council called Engineering Life. This project is really at the heart of it. We're interested in better understanding the relationship between engineering and biology today in this kind of contemporary age. The core research question at the heart of our project is really to think about how engineering ways of knowing and engineering ways of doing are beginning to transform the ways that we work with biology. And as a team, we're exploring this from several perspectives. We're thinking about it conceptually, so engineering ideas and conceptually how they interface with biology, thinking about some of the promises, visions, and aspirations for the field, really engaging at the level of practice, understanding what's happening on the laboratory floor, and thinking also about some of the larger policies that are framing and shaping this relationship between engineering and biology. This is a really large project team. It's a very large kind of social science project led by Jane Calvert out of the University of Edinburgh. And the work that I'm going to talk about today is really some ideas that we've been bouncing around. There are 4 of us on the team who are quite interested in automation. And for the last year or so, we've been building up some ideas around this. I'm going to flag up explicitly at the end a little bit of the work that Chris and Annie, who are 2 of the graduate students part of the project, really interesting work that they've been doing around this topic as well. So my own research interests really revolve around the governance of emerging biotechnologies. And by governance, I mean quite a broad range of sort of formal and informal mechanisms by which we shape the trajectories of our technologies. And we make decisions about what they will, the kinds of directions we want to take, the kinds of outcomes that they will have. One of the ways that I'm really interested in this is by actually looking at the everyday practices of biological engineers. So sometimes the more informal ways in which we sort of shape the trajectories of our work. And I'm really interested in infrastructures that support or that are designed to enable biological engineering. So I've done a lot of work around standards and protocols, and over the last year or so have become more interested in some of the larger physical and digital platforms. What I'm going to talk about today mostly is focused on DNA foundries and the kinds of biofabrication facilities that are emerging to enable biological engineering. So sometimes I get these interesting questions. It seems people are a little bemused by the fact that infrastructure might be seen as a sociologically interesting topic. So I'm just going to kind of try and share with you very briefly a take on infrastructure and why it might be interesting to study from this sort of broader sociological perspective. So one of the things that, to start off with that I find interesting about infrastructure is that it's not neutral. Infrastructure is a world-building activity. You're going out there and physically building your hopes, your aspirations, your ideas about how to work into material form. And I think that's something quite profound. We know too that building infrastructure involves making design choices. And this is something as experimentalists, very, everybody knows this, right? You're choosing, you want to ask a question, and you know that the equipment that you choose to use, the way you set up your assay, is significantly going to affect the type of question you can ask, the type of data that you're going to get out of that assay, and what you can do with it. And the same holds true when you're talking about larger scale infrastructure, but perhaps the consequences of it become more profound because you're investing a lot of time and resource into building that infrastructure. So you might increasingly lock yourself into particular ways of thinking. So these choices really do shape the range of possible outcomes that you can achieve. In the early phases of emerging technologies, there's often a lot more explicit discussion about infrastructure. So sometimes there's resources being made available to construct new platforms. We sometimes get contestation or debate or discussion about the kinds of design choices we should be making. And from a sociological perspective, those are really interesting ways of trying to unpack and understand some of the values and visions that are driving, this push to develop infrastructure. In that sense, I think also we can think of infrastructure as a powerful form of governance. When we build infrastructure, it begins to shape what we can do and has significant impacts on the downstream trajectories of our technologies. I've put here a form of self-governance because I think oftentimes infrastructure, the discussions and debates about how to do this are often very contained within a small group, usually of technical experts. So in that sense, too, these decisions that shape the trajectories of our field are being are often taken by a small group of people. So as I said, today I'm going to talk about this phenomenon of biofabrication facilities or DNA foundries or biofabs. They take on several different terms. This kind of growing phenomenon. This is something that was picked up a year ago by The Economist, talking about these robotic labs for high-speed genetic research, profiling some of the foundries that have been established. And I just the other day tried to pull together, this is a very partial list of foundries that have kind of in the process of being developed over the past decade or so. They're appearing in the US, in Europe, in Asia, in academic settings, in industry contexts. I've put the JGI on this list. Obviously, the JGI is one of the older facilities. It's not, it's 20 years old. But it is constantly innovating in this space in terms of the services that it's providing. And when we talk specifically about some of the DNA construction, assembly, and cloning techniques that are at the heart of a lot of these foundries up here, the JGI's strategic goals are certainly moving in that direction from what I can see as well. So a lot of interest recently in building these biofabrication facilities. And this prompts a number of research questions. What is it that these facilities are bringing to the table? Why is there so excitement? What can they contribute to biological engineering? So a couple of the questions, the research questions that we're starting to ask and do some research around include, how is value being imagined and embodied and created through these facilities? And then once we have a handle on that, trying to think through some of the emerging implications for the future of biological engineering. So I'm going to share with you today just some early ideas in this kind of along these lines about value creation and about what that might mean for biological engineering. The way that we can do this, in keeping with the Engineering Life project, is to look at visions, aspirations for these foundries, to engage with what's happening actually on the floors of these facilities, and to think about some of the policies that are shaping their creation. So when we look, in terms of visions and aspirations, one place you can turn to try and get a bit of a handle on this is some of the kind of wider policy and strategic documents around biofoundries. You can look at the front-facing materials, so the websites and the publicity materials that they put forward, news stories about them, some of the research publications that are coming out that are making use of these facilities, and so on, to try and understand what's there. So there are a number of different visions and aspirations that appear. They're interrelated, but they're all slightly different. So I'm just going to put a few of them up. This isn't an exhaustive list, but to give you a sense of how they're being talked about. So we get a lot of discussion about the scaling up of genetic construction and testing. And this scale up is linked to ideas of efficiency and speed, so we can build greater numbers of DNA constructs faster, more efficiently, more reliably. There's discussion about human error. So if we're going to try and increase the throughput, human error is a natural part of kind of laboratory work. And for those facilities interested in automation, this might help to reduce human error. Sometimes this is plugged into discussions about the reproducibility crisis, which seem, kind of are much bigger than just engineering biology at the moment. There's a set of really interesting and I think underdeveloped discussions around the role of human labor in these facilities. So are we talking about replacing human labor with machines? There's talk about de-skilling the kind of work that's needed in order to construct DNA. There's also talk about maybe not replacing, but kind of shifting the type of work that gets done. So substituting the donkey work of genetic engineering with allowing instead researchers to focus on the design, so giving them a little bit more creative freedom in the process. A set of ideas here that I think feature but haven't been fully articulated. There's also, when sort of in the outward-facing material about these foundries, explicit discussion of some of the broader societal goals that they're trying to achieve, often described in terms of revolutions, which is interesting language to use. Revolutions in biomanufacturing, so a shift maybe from more artisanal biology to industrialized biology, a sense in which these foundries are positioning themselves to be disruptors of existing high throughput biotech in the form of ag biotech, pharma, chemicals, and so on. So the ways in which they are doing that is something worth kind of looking at in more detail. And we often see this plugged into discussions about sustainable biomanufacturing as well. So enabling the exploitation of genetic resources to foster or catalyze a shift away from a petroleum-based economy. So this is just a kind of a snapshot of some of the visions that are at play, and you can see they all kind of, they're approaching, they're tackling the issues from slightly different angles. Some of them may be more or less compatible with one another, but they all feature in this space. So if we start to look at what's happening in terms of building these foundries in practice, we've begun to make contact with and make visits to both academic and industry biofoundries. And across the board, we find that kind of for the foundries that we are visiting, there's a common shared interest in building capacities to scale up DNA design and construction. So particularly when we're talking about DNA assembly and cloning, a little bit not necessarily straightforward synthesis, but these kinds of larger constructs where you're trying to build multiple variants in order to test different pathways and so on. So there's this shared interest. But there are many differences in terms of the details of how this is being pursued. And I think that's really interesting because that links to the value propositions of these facilities. So to flag up just a few differences of, that we've encountered so far, different degrees of investment in high throughput robotics and software. So an interesting question about the degree to which you feel that your foundry is dependent on having the most cutting edge high throughput technologies that you're embedding and incorporating, or the degree to which you might say, well, we're going to focus on the technologies that we already have. They may actually be much lower throughput, but we're going to develop the processes, the protocols, and really focus on trying to standardize the work that goes in to improve the reproducibility of the facility. Accompanying this, different views or philosophies about how you might configure and articulate the workflows in your facilities. So do you believe that, that there is more to be gained from having modular work cells where people might be involved in moving samples from one bench to another? Or are you trying to develop a fully automated, integrated system with robotic arms that basically gets the samples from the fridge and gives you an output at the end? This is also linked again to different imagined relationships between humans and machines. So across the different foundries or facilities, we see kind of subtly different arguments being made about whether the goal is to substitute human labor, reduce the need for people, versus really trying to augment or optimize human work. So really thinking about, well, what is it that machines do very well? What is it that humans do really well? And can we really try and balance that out? So these kinds of differences, I would argue, are salient for how the value propositions of these facilities are taking shape. So I just wanted to talk very briefly about kind of a handful of implications. So not just how are these facilities being constructed, but what does this mean for the future of biological engineering? And the first implication, I tend to think of in terms of scaling down to scale up. I'll try and explain what I mean by that. So, the idea being that essentially, if you're really focusing on high throughput automated technologies, there's this push to get to increase sample size and to decrease sample volume, right? And so moving from possibly the Eppendorf tube, or at least the 96 well plate, down into these phenomenally kind of rich, 1,500 well plates or more, right? But the process of scaling down isn't necessarily trivial. So there was a short paper that came out a couple of years ago talking about the challenges of translating assays from tube form to a microplate format. And these challenges, I think, are both technical and conceptual. So from a technical perspective, when you're scaling down from milliliters to microliters, in this case to nanoliters for these 1500 well plates, you're fundamentally changing the reaction kinetics, which means you have to redesign the assays around them, right? So there are these technical challenges. And there are also conceptual challenges for researchers who haven't necessarily been working in this space. Certainly in my PhD, I use 96 well plates all the time for cell migration assays and for ELISAs and so on. And if you suddenly told me I could conceptualize my experiment in terms of 1,500 samples, it would take me a while to understand how that, how to actually think about my experiments in that way. So there are very few sociological studies of automation that have been done, but there's one from 20 years ago looking at the introduction of DNA sequencing. And they said at the end of their study that automation is actually not just about the mechanization of protocols, but actually fundamentally reconceptualizing them. And I think this kind of challenge is at the heart of what a lot of these foundries are currently trying to do. And reconceptualizing these practices then has potential long-term implications for how we think about experimentation even, right? So I think in some cases what we're seeing is a shift from thinking about the experiment or the project as your base unit to really kind of shifting your focus and thinking about, well, how do we keep the machines busy? How do we optimize their performance, right? That's actually that's a potentially long-term profound shift in how we conceptualize biological work. One of the kind of ways that we're seeing this emerge in these kinds of high throughput facilities is the notion of batching that we hear as a way to basically make most efficient use of your machines. So you may be stacking projects together and so on with the aim of keeping the robots appropriately busy and being able to move your samples across machines in an efficient way, right? This is a reconceptualization of how we do work. Another set of implications I think that's emerging that's really interesting and worth thinking about relates to some of the priorities and trade-offs that are at play. The kind of examples here that I think are really salient revolve around workflows, right? This is a term that has come up several times over the past couple days. The idea of a workflow really signaling an attempt to try and rationalize your practices. And in synthetic biology at least, and engineering biology more broadly, one of the kind of key workflows that we hear about all the time is this design, build, test cycle. And then within that, you can develop a lot more granularity as you actually try and push your work through this kind of workflow or structure. So, but there are inevitable trade-offs when you're trying to develop workflows. And this is something that kind of emerged for us during a workshop that we held at ASU a couple of years ago, where we brought together a group of practitioners in academic and industry facilities, and we prompted them over a course of the day to map out workflows that they were developing in their facilities. And we were mapping out workflows side by side and doing comparative, doing some interesting comparative and troubleshooting exercises. And there are just a couple of takeaways from this that I want to make today. So one was that we had at one point we had two very similar workflows in a sense being mapped out side by side. So two industries that were mapping out the workflows that they had developed to do sequence quality control. And actually the workflows looked reasonably different. And it turned out when we tried digging into why and how they looked different, that they were aiming for kind of different long-term goals. One of them was aiming for scalability in terms of the number of sequences that they could push through this quality control pipeline. And the other one was really oriented around flexibility in terms of thinking through the diversity of types of sequences that it might be able to accommodate. Became a really interesting trade-off, right? Both of those arguably are, broadly speaking, under the heading of efficiency, but efficiency might mean different things. And I think we have to remember, too, that even the idea of a workflow is a particular way of representing your work. One of the historians at this workshop joked that it was a morality tale about efficiency, but I think there's something to be said for stopping and thinking about how we're representing our work. And in almost all of the workflows that were kind of mapped out during this meeting, people didn't feature in them at all. So it was really about the transition of information and digital samples and so on from one place to another, and really didn't think about who was doing that work, what kinds of skills and expertise they needed to have. Which is the last kind of the sort of set of implications that I wanted to flag up for this particular talk. Thinking about skills and expertise and what we need really to be able to enable this kind of successful high throughput automation. So the first idea that I want to raise is based on work that Chris Mellingwood has done. So he's just graduated with his PhD from the University of Edinburgh, and as part of his research, he spent an extended period of time in 2 different academic foundries in the UK. And what Chris was doing was he was really shadowing the scientists and the engineers who were in charge of setting up and managing the automated workflows in these 2 facilities. And he was really trying to get to grips with the kind of expertise that they needed to have in order to be able to do their work effectively. And what Chris came up with, I think, is this really fun idea of the amphibious researcher. And so amphibious, in this case, coming from the Greek word meaning to lead a double life. And particularly appropriate, I think, in terms of what Chris was talking about, was this need that amphibians are able to live and to adapt to both wet and dry, contexts. And so what Chris was observing was the real kind of skill set that this particular group of practitioners had in terms of being able to integrate both the dry lab and the wet lab practices to achieve successful automation. And he sort of described this amphibious working knowledge as being absolutely critical to getting your pipelines up and running. One of the things that he highlighted was that it's possible to get your robots to work, but that you actually have to learn how to get them to work with the organism. So particularly when you're putting kind of biological samples through, it's not good enough to say that they work. They actually have to attend to the particular needs of the organisms that you're working with. And so I think this is a really fun kind of idea and way of thinking about the expertise that's required in order to successfully automate biological platforms. Annie has been doing some work in industry facilities in the US. So she's begun some pilot work, and she's planning to do some more extended visits to different industry facilities. And she's very interested in this idea of workflows. And I'm going to share with you one of the observations that she has come up with so far in her study as well. So she has been asking practitioners at the facilities that she's been spending time in to draw out their workflows. And she's been struck by their kind of very linear presentation. And it turns out that most of the time, this linear workflow that is being sort of drawn out for her really involves the tracking of digital information, right? And that when you start feeding biological samples through the pipeline, the work starts to look a little different. So she's been following, again, the practitioners who are making this work happen, and noting that there's in some ways a predictable unpredictability to kind of achieving the kind of this workflow. And she's been highlighting all of the work in a sense that needs to happen kind of around and under this seemingly linear workflow to make it happen. And she's been calling this phenomenon kind of routine interruption. So when you start feeding biology through this pipeline, it may be that it's not able to be rendered in quite such a rational form as those boxes describe. And there's a lot of creative work that's needed in order to sustain the appearance of this linear flow, right? And actually, the skills required to sort of execute this very elaborate choreography to kind of maintain this linear flow is something that can take years to achieve. So in the kinds of interviews that she's done and the shadowing that she's done, for a mature operator who's able to seamlessly work this kind of workflow can take anywhere from 3 to 5 years. So again, I think this raises some really interesting questions around the expertise that's needed in order to successfully be able to automate and manage these workflows. So just summing up, I hope that this talk at least has been a little bit of a provocation to start to kind of begin a conversation about the types of infrastructure that's being built, and to acknowledge that any infrastructure, and particularly the infrastructure that we're currently building for biological engineering, requires a really complex integration of mechanical, digital, biological, and human systems. And it's actually the details of those configurations that start to matter, right? We can talk about it at this high level, but when we look across the large array of foundries and facilities that are currently under development, it's the specific configurations of these that are helping to articulate their unique value propositions. And I think that these configurations, how they manifest themselves, have implications in the long run for how we think about experiments, for the kinds of trade-offs that get built in and then cemented into these workflows, and the kinds of long-term consequences this might have for the field, and also both in the short and long term thinking about the skills and expertise that are needed to maintain this kind of work. So this is just kind of, as I said, some preliminary work that we've been doing. The Engineering Life project is in the process of wrapping up, but I have a new collaboration underway at ASU with Gaiman Bennett and Annie, who's been doing some of this pilot work in industry foundries. And what we're planning to do over the next couple of years is a more detailed comparative analysis of biofabrication facilities in the US, focusing on both academic and industry facilities, and really to dig deeper into a lot of the questions that I've raised for you today. So looking at their emergence, how they're articulating themselves in relation to more established biotech, how they're building their value propositions, and really also attending to the experiences of the practitioners as they go about this, I think, significant challenge of trying to sort of automate engineering biology workflows. If any of you are interested in this work, would like to collaborate or have questions, I'd love to chat. So thank you very much for your time and attention. I'm happy to take any questions.
So I'm just curious, in any of your discussions, was it ever considered that automation is not just inevitable, it's not necessary? And so there's no choice, you have to move there. So if automation becomes necessary or inevitable, and there's no choice, and so then irrespective of all of these other parameters, is there ever a discussion about what human beings can now do that they couldn't do before? Not so much to take away our jobs or alleviate our donkey work or whatever, but all of that is necessary. It's gone. Machines are doing it. Fabulous. What do we do? Write poems, make paintings.
So I think a couple of quick responses. I would really resist the language of it being inevitable, because automation won't happen unless there is significant investment in it. It is not suddenly going to appear, right? So it is a choice. It might be a widely accepted choice, right? That this is something we're going to pursue, but it really isn't inevitable. That being said, there's a widespread enthusiasm for it, which might make it seem like it's inevitable. I think, interestingly, so in the public discourse, kind of broadly speaking, not engineering biology, we get a lot of this. Annie actually describes it as two competing visions. You get the robot apocalypse vision, where the robots are going to take all of our jobs, or the techno-utopia vision, where the robots do all of the drudgery that we don't want to do, and we're freed up. So this idea of being freed up to do creative work. And the answer is clearly going to be somewhere in between. Interestingly, in the synthetic biology space, I don't think that conversation is really happening. I think there's a sense in which there's so much work needed right now just to make it happen that the kind of longer term we're maybe not talking about too much. Anyway, happy to chat more about that. Yeah.
Sorry, time's up, actually. Thank you very much, Emma, for the nice talk.